utåt.
So, start again. Sorry about that. I was mute. So, good evening and welcome to this second night of the Pint of Science Festival in Tromsø. We, the volunteers of the festival, Fiorida, Christopher, Linda, and me, Jaume, are very happy to be back. For those that did not join us yesterday, Pint of Science is an international science festival aimed to bring scientists to a pub to explain their research to everyone. This year, unfortunately, we are going online. And I'm sure that you know the reason, right? But this situation is not going to take away our motivation to learn about new topics. Today, we're going to talk about DNA. What type of DNA? Sandra Garces-Pastor is going to talk about environmental DNA. Then, Peter Heintzman is going to tell us about prehistoric fauna, horses and mammoths, which is called ancient DNA. So, don't lose more time and let me invite Sandra. She's a postdoctoral researcher at the Arctic University Museum of Norway. Sandra is an expert on paleoecology, ecosystems, and DNA analysis. She's a member of the EcoGene group, and I'm sure she's eager to start. So welcome, Sandra, and thanks for being here with us in the Pint of Science. And if you're ready, the screen is yours.
Thank you very much, Jaume, and thanks for the opportunity to talk about DNA today. So today I'm gonna talk about how we can use the DNA to better understand the biodiversity. To do so, I will explain several examples regarding several animals to explore what we can learn through DNA. As you know, all the cells are composed of DNA, and as you have seen in many movies and series, we leave traces of DNA everywhere. So we can find DNA in our skin, so in skin cells, in nails, in hair, in blood. There's practically everywhere. So basing from this, there are a lot of efforts to sequence all the biodiversity of the world. So all the living organisms that are in the world are being sequenced so we can extract the DNA and we can focus on small fragments of this DNA that allow us to differentiate between species. And with all this data, we can create a reference library where we are storing all those sequences of all the living organisms. This is huge because we are talking about bacteria, plants, animals, everything. And to do so, we can use several DNA fragments, like, for example, for plants would be chloroplast, and but also mitochondria, while for plants it would be mitochondrial or nuclear DNA fragments. So from now on, I will talk about these barcodes, and we will focus on several organisms. Why it's so important to have all this biodiversity sequenced? So, for example, because if you go to the field and you find an insect, you say, well, I already see it, and if I use a taxonomy, I can describe which insect it is. Like, for example, here we have two organisms, but what if you find just one leg? You cannot describe which organism is coming from, but if you extract the DNA and you compare your sequence with all these DNA fragments from this reference library, then you can know which species we are talking about. So here I want to show you an infography that I think it englobes very well, but it's like a marine ecosystem. So we already know that we leave traces of DNA everywhere. So do the animals. Like, for example, this whale, it's leaving a lot of traces through the tears or the saliva or feces. But the same, it's for the tuna, that it's and also for the fish that this tuna is feeding on, or the dolphins that can also live in the same region. Not only the big animals, we can also go to the small invertebrates like the sea urchins or the sea ears, that they are also living DNA traces. And also we have other organisms like bacteria that come from the cities or from the waste from cities. And we can find a mixture of all this DNA in these ecosystems. So according to that, we could take a water sample of this place without any of these organisms inside, but we could find DNA from those organisms, right? So here you have the first example, and up there you can see a typical freshwater ecosystem. So you have plenty of plants, animals, and some of those animals are aquatic, so we have a lot of fish. So when people want to study fish, they do fish surveys, and they might recover part of the fish that you can see here. So probably smaller fish would properly hide, so they won't be able to catch them at the first time. And so they will need a lot of times and repeat that fish survey during several times to get an idea of the whole fish community that it's living in that ecosystem. So my first example, it's about estuaries from 3 places of the UK. So what they did was recover water, like the equivalent of 2 pints of water, of these estuarine areas. And they were looking for animal DNA, and most concretely for vertebrates. They were looking for vertebrates and for fish. So when they sequenced that DNA, they found 93 fish species, which is a very good result and a higher amount of fish than a normal survey. But they could also find 32 species of birds and even 8 species of mammals. Not only this, when they take a closer look to all the birds that they found, they realized that some of those species were birds that are considered as a conservation concern, which are not endangered, but they are close to. One of the cases also it's the spoonbill, which is an animal that hasn't been found there for many years, and only recently this bird came back to the region. So you can see how sensible this technique is. But also, these researchers had a surprise because we are talking about the strains from the UK, and they found DNA sequences that come from buffalo and 2 different species of kangaroo. And as you know, in the UK, there are not many kangaroos. So the first question that arises is like, okay, maybe we have contamination. And they check all the quality controls and proceedings that they did in the lab. But everything was fine. But they realized that just 2 miles above one of those estuaries, there was a meat processing factory of exotic meat. So the DNA of those animals end up in the lake and then in our water. I will get to another example, and it's the brown shrimp. So there were some researchers that were studying the cycle of this shrimp, and what they wanted to know is what this animal was feeding on. To do so, they took some organisms and checked the stomach content, but they also took samples from the water where this organism was living on, and also the sediment. And when they extracted the DNA, they realized that the 3 kinds of samples were telling different stories, but that in general, the stomach contents could get much of the information that the other samples were giving. So in a way, by taking the gut contents information, you could have an idea of the animals that were surrounding this shrimp. So in a way, that shrimp was acting like a deep bottom sea vacuum. It was eating all the animals that were ending up at the bottom of the sea. And then by getting those gut contents, you could know which are the animals that were living there. Not only this, what they could also see is that when they checked closely which animals were they could even see the trophic relationships between this shrimp and his environment. So for animals that were smaller than the shrimp, you could tell that the shrimp was being a predator. There was a predatory relationship, so the shrimp was hunting them or actively eating them. However, other animals that were much bigger than the shrimps, it was impossible that this animal was hunting them. But anyway, that could be a scavenger. So the shrimp was feeding from all these bigger animals that end up at the bottom of the sea, and he could reach to them. Not only this, if we change to another kind of shrimp, the pink shrimp, it's closer to here, to Tromsø. They did a similar study using the gut contents of this shrimp, and also they wanted to compare it with the traditional survey where they used these troll nets at the bottom of the sea. And here up you can see the two kinds of results that they found. So here you have the troll and the 12S, that it's the barcode, the fragment of DNA that they use to amplify. And each color, it's where those animals live. So cryptopelagic would be animals that live very close to the surface, while pelagic live in the water column. The demersal would live in the deeper water column, and the benthic animals would live directly in the sediment. And what you can see at first sight is that with the troll, you have a very low representation of all these organisms that are living in the water column. So as you can see, what the shrimp was eating and what you can find in its gut contents, it's a better representation of the ecosystem where this animal was living on. So in a way, this organism was acting like a natural sampler. Then we have seen the example of with the estuarine study of filtering the equivalent of 1 pint of water. But then other questions arise. What would happen if we use animals that can filter 100 pints of water, such as sponges? These animals are feeding from all the organic matter that is floating and small animals that are floating on the water. And it's filtering full time, 24 hours a day. So some researchers went to the Antarctic and took some sponge samples and extracted the DNA. And apart from the DNA from those sponges, they could also find DNA from vertebrates like seals, penguins, the fish that those seals are feeding on, invertebrates. So whole ecosystem that was surrounding those sponges, which you could not survey doing a traditional survey. But this not only stays here. Some people also studied some sponges in Vietnam, and they sampled 64 sponges of different species, and they could find 90 fish species. And only with this sampling, they already covered 1/3 of the traditional conventional surveys, which is quite a lot. The main fish that they found were the fish that typically live in the reef, in the coral reefs, and they are the ones that are closer to the sponges. So these were the more expected to find. Not only this, they also found these pelagic fish that live in the water column, a bit more apart from the sponge, but they were nearby. But what is most surprising is that they could also find sequences that belong to deep sea fish that were quite far away from that sponge. Not only this, they could also find migratory fish that were passing by in that in that period. And we know that sponges are sessile animals. They live in one stone and they filter plenty of water. But then more questions arise, isn't it? What if we use animals that are also filtering, but they are also floating? So they can also give us another idea of the environment that surrounds them. And that would be another vision of marine biology. Not only this, we also have jellyfish that they are also floating. They don't filter, but they are sticky and they feed off some animals. So we could also have many information from these floating natural samplers. Now I will get to the terrestrial land, and many studies that or former traditional surveys of terrestrial biodiversity was to put hidden cameras in several parts of the forest or other places, just to see which animals are living in there, because some might have more nocturnal habits or are try to avoid humans and everything that could smell like human related. And this is cool, but sometimes you only find one situation, like one scenario, one visualization of one animal, like just for example, this bobcat. And it's very nice to know that this animal is living in the territory, but it's also nice to know what do they feed on, how many are there, and to apply proper ecological measures to preserve them. But there's another way. Instead of using hidden cameras and wait to see the animal passing by, you can also use other animals that feed on them. And in this, we can use like all these blood feeding animals such as leeches, mosquitoes, or ticks, that if any animal, any vertebrate pass nearby them, they will jump to them, feed a bit on them, and when they had enough blood, they will come back to the plants that are on the floor to digest that blood and wait for the next prey. But this gives us a chance to go to that region and take all these animals, extract the DNA, and we can see all the vertebrates that also live in the terrain. It's like an indirect way also to have this idea of the biodiversity that is in that. And continuing with some invertebrates, I'm going to talk about the pollinators, that not all of them are only arthropods. We also have some vertebrates and bats and birds. But today, it's the World Bee Day, and I'm going to tell you one of the studies that is related with pollinators. This is a study that was done in Denmark, and what they did is they sampled 56 flowers of 7 different species. And as you can know, all the arthropods and everybody leaves traces of DNA. So if you go to a flower and extract the DNA, you might find DNA from arthropods who visited those flowers. They found 135 arthropod species, not only pollinators, but the parasites of some insects, the predators of these pollinators, some predators of the plants itself, and the animals that were feeding on those plants. But this not only stays here, you can also look at the opposite way. So who visited each plant, creating a huge network between plants and arthropods. Also, we have said that there are some animals that are critically endangered, and it's very difficult to sample them. But because the populations are so endangered, it's very important to know their biology and what do they feed on to be able to provide the resources that they need to continue. So I'm going to go to this study that it's about the Montseny-broke newt. It's a very critically endangered salamander that lives in Catalonia, in the Iberian Peninsula, just in one natural park there. Population, it's really, really endangered. So they wanted to check if they can if they can see what this animal feeds on, and to be sure, so animals feed on what they predate, and then at some point when it goes out through the feces, and if you get to those feces, you can extract the DNA, and then you might know what they were feeding on. They wanted to test if you could tell that from this newt. So they took two animals and feed them apart just to know if they could retrieve information from the feces. So they give him just one kind of food, that is the small crustacean, Artemia, and then they waited until it went out. And what they found was that in the feces there was a lot of Artemia DNA, which makes sense, some DNA from this newt, but also plenty of DNA of fish. And when that was a very surprising result, but when they realized something, is that the Artemia was being fed on this fisheries waste, like all these fish were pulverized and given it to the Artemia to feed on. So by using the DNA from feces, you could not only know what the newt was feeding on, but also what the Artemia was feeding on also, which is a very sensitive technique. Not only this, and I will jump to the bats. And here they check, so you have several bats, you want to know what they are feeding on. And then they took the feces also, and they amplified. And what they could see is that they were feeding on different kinds of arthropods, so they were not completely competing for the same food, which is good and it helps for the biology of those species. But they also realized that a big amount of those taxa, those insects that they were finding, were actually agricultural pests and insect disease vectors. So these bats were suppressing all these pests that were threatening crops and also human health. And finally, I'm going to talk about the soprano pipistrelle, that it's a bat that lives also in Catalonia, in the Iberian Peninsula. And, and they wanted to know what these animals were feeding on in a region where there are a lot of rice crops. And what they found in the feces of this bat was the first record of rice water weevil. This insect, it's a pest that comes from North America and arrived to Europe via Greece, then Italy, then France, and eventually it had to arrive to Catalonia and the Iberian Peninsula. But the difference is that the first record came from the feces of a bat, and that is good because then farmers can know that this organism is around the crops, and before this organism multiplies to a level that it can become a pest and a threat for the rice crops, the farmers have time to prevent this. With all these examples, I wanted to show you which is the potential of the environmental DNA to study biodiversity, since it's a very little destructive technique, and it's more efficient than the classical surveys. And this allows us to reach to species that are very difficult to monitor because they are endangered, because they are very difficult to reach, or because they are very shy. But anyway, this opens a new phase for ecological studies. And with this, thank you very much for your attention.
Thanks, Sandra, for this inspiring presentation. It was really great, and we really enjoyed it, so thank you. And for the audience, remember that you can ask questions in the chat on the YouTube channel, and our speakers are going to answer them at the end of this event. And now comes Peter to talk about prehistoric fauna. But before, if you are enjoying the festival, please click like on the video, and if you love us, subscribe to the Pint of Science Norway channel. You will make us very happy. Peter is an associate professor at the Arctic University Museum of Norway and member of the EcoGene group. He is an expert on paleogenetics. So please give him a warm welcome and thanks for joining us. If Pete, are you ready? The screen is yours.
Yep. Great. Thank you very much, John, for the invitation, and it's great to be here. Okay, so I'm going to talk to you today about the hidden diversity of extinct megafauna and the role that ancient DNA is playing in basically revealing this hidden diversity. Okay, so let's just introduce the megafauna. So within the last 2 to 3 million years, nearly every habitable continent on the Earth was covered in megafauna up until around 10,000 years ago. And these megafauna ranged from elephants that you would get in the tropics to elephants that you would get in the Arctic, like mammoths. You also had giant ground sloths that lived in South America, and you had these huge armadillo-like relatives called glyptodonts, which were actually the size of small cars. And even in the far north of the UK, of the planet, in northeast Siberia and northwestern North America, we have megafauna. There were mammoths, woolly rhinos, horses, bison, bears, all sorts of weird and wonderful animals. But if you go to these places today, you don't see any of them. In fact, there are very, very few of those megafauna actually survive today. Examples include reindeer and moose and some others. But for the most part, the vast majority of the megafauna that existed for millions of years is now extinct. So why is this? What happened? So there are kind of two competing hypotheses as to what happens. First is, and these are the main hypotheses, the first is that the megafauna were just simply hunted to extinction by people who, when the megafauna went extinct around 10 to 12,000 years ago, these peoples were beginning to expand around the Earth. The second kind of key hypothesis is climate change. So, and there were big swings in climate change happening at this time. So it's really hard to disentangle these two causes. Potentially, it could be a really complex interplay between these causes that actually cause these extinctions. But really, to kind of get at why the megafauna went extinct, before we kind of get at that question, one of the key things we need to understand really is actually what went extinct and how extensive were these extinctions. So we can obviously say woolly rhinos went extinct and woolly mammoths went extinct, but how many distinct population extinctions were there? How many of these populations were really ecologically key to their ecosystems? So basically, how severe were these extinctions ecologically? And the way we get at this is by looking at bones. And one of the best places to find bones of extinct megafauna is northeast Siberia and northwestern North America, where bones preserve extremely well in what we call permafrost, perma-frozen ground. These deposits are absolutely fascinating. They're full of bones, and in northeast Siberia, people basically try and find the bones and other remains that they can sell. And in northwestern North America, these bones are washed out of the permafrost as people hunt for valuable minerals such as gold, as is shown in the top corner here. And there are lots of bones that come out of these deposits. So this is just basically a collection of megafaunal bones that have come out, that have just been collected in a single day from a single locality. Not only is the permafrost great at preserving bones and their anatomy, their morphology, permafrost is actually really great at preserving the DNA within them, the ancient DNA within them. And it's the ancient DNA that we can really use to examine how populations have changed through time. But first, let's just take a step back. What is ancient DNA? So, as you all know, DNA is found in basically all living cells, and it's really, really long. So the longest DNA in your cells is actually around 1 million times longer than the cartoon that I'm showing on the slide here. Really, really long. But then rapidly, after death, the DNA fragments. It becomes really, really short to become basically as short as the molecules that are shown here. Not only does the DNA fragment to become very short, the ends of the DNA also become modified. They become what we call damaged. And these damage signatures that we see at the ends of the molecules are how we characterize ancient DNA and basically authenticate that it's real. DNA preserves in several environments very well. It's good preservation conditions are cold, dry, and stable environments. So permafrost is perfect. Also, caves are really good environments. And bad environments for DNA preservation are basically those that are hot, wet, and have a lot of fluctuating temperature. So for example, the tropics and deserts. And because the ancient DNA is very fragmented, it's also very low concentration relative to kind of like modern living DNA. When we're dealing with ancient DNA, when we're sampling it and we're doing our thing, we have to make sure that we basically dress up in this kind of as like crime scene investigators to basically protect the specimens, the ancient remains, from ourselves, because we are the source of contamination. So how does a kind of like a methodological workflow look for this? So here's just a quick mock-up. So we start off with a chunk of bone that we might have just drilled out of some remains. We then extract the DNA from it. We end up with this little bit of clear liquid. We hope there's DNA in there. And then we sequence that DNA. Then we perform some bioinformatic analyses, which is basically a way of sorting out the DNA sequence data to make sense of it. And then we can analyze those data and build what we call phylogenetic trees, which are family trees, to look at the relatedness of these ancient populations or ancient species to living species or other ancient populations. So what has ancient DNA taught us? So I'm going to just give you two kind of like case studies, basically one on horse, one on mammoth. So I'll start with the horse, and I'm going to tell you the story of the North American stilt-legged horses and basically how ancient DNA resolved what these things were. So wait a minute, what are stilt-legged horses? So let me back up a little bit and just tell you a bit about, just a bit of basics about the modern day horse family. So horses, we have horses, donkeys, and zebras. In total, we have 7 different species, with zebras and donkeys more closely related to one another than horses are. So horses are what we call the outgroup to donkeys and zebras. But in the Pleistocene of North America, we have lots of different species that have been named, and how valid these species are is up for debate. But there are 2 major groups. There are other groups known, but the 2 main groups known from the fossil record are the stout-legged horses, shown here in this blue, and the stilt-legged horses, shown in the orange. And the way we tell the difference from them, as the name suggests, is the stout-legged horses have these much broader foot bones, these limb bones, whereas the stilt-legged horses have these much thinner, more gracile limb bones. So these are very easy to tell apart in the fossil record from the leg bones, although not necessarily from other parts of the skeleton. Based on the anatomy, the morphology of these bones, stilt-legged horses have been thought to be closely related to the Asiatic asses, the donkeys from Asia, Equus hominis and Equus cayenne. So based on the anatomy, stilt-legged horses belong with these donkeys. However, the stout-legged horses, they're very, very similar to living day horses, Equus caballus, and so they've been placed next to them. However, the first kind of like ancient DNA studies were based on basically just very short pieces of DNA. We're suggesting that actually the stilt-legged horses were more closely related to regular horses and not to these Asiatic asses. So, which is correct? Is it the small amount of ancient DNA, or is it the morphology? And this is the question we wanted to get at to really understand what these stilt-legged horses were. And the approach we used here is we used remains that were found from across North America to see if there was any variation. We sampled multiple bone types, foot bones, leg bones, and even a skull. And unlike the previous ancient DNA studies, we targeted multiple parts of the genome to give us a much fuller representation of what, and robust representation of what these stilt-legged horses were. So this was the previous ancient DNA-based tree, but based on our findings, actually what we found is that the stilt-legged horses fell outside of the diversity of living horses. So this is very interesting. This is very different to anything that was previously thought based on either anatomy or previous ancient DNA studies. And because this fell outside of living horse diversity, which can be interpreted as the genus Equus, we actually gave these stilt-legged horses their own generic name, genus name, which we called Harringtonhippus. Now, that's all very well, but what's very interesting with this is that the Harringtonhippus, like the stout-legged horses of North America, went extinct around 10 to 12,000 years ago at the end of the Pleistocene. Now, if you had thought that stilt-legged horses fell within the diversity of living horses, what that would say is that when these horses all went extinct in North America, the kind of like total diversity within Equus would have been basically maintained, because we still have donkeys, zebras, and regular horses. But because we now know that stilt-legged horses fall outside of that diversity, it basically makes us appreciate that actually the losses in North America were actually much greater. And there's actually another genus of horse that lived in North America, but was mainly found in South America, Hippidian, which is even more different, and that also went extinct at this time. So we're basically losing multiple genera from the Americas at this time. Okay, so that was the horse story. Let's switch gears to mammoths. So I'm going to talk about basically a paper that we published just a couple of months ago, where we recovered some really, really old DNA. I didn't say in my introduction how old the oldest DNA possibly can be. Got bad news. We cannot get DNA from amber. We cannot get dinosaur DNA. DNA does not survive for tens of millions of years, unfortunately. It'd be great if it did. We'd love that, but unfortunately it doesn't. The oldest DNA that's been authenticated, that is on record, was our study from 2 months ago, where we recovered DNA from Siberian mammoths that were up to about 1 to 1.2 million years old. So just a bit of background about mammoths. In this talk, I'm just going to talk about 4 different species, or 4 different taxonomic units here. We've got the southern mammoth in the top left. This was the earliest of these 4 mammoths. It lived around 3 to 2 million years ago in Eurasia, in the southern parts of Eurasia. Then, around 1.7 million years ago, the steppe mammoth evolved from the southern mammoth and lived until around 200,000 years ago. And the steppe mammoth is known from both Eurasia, northern parts of Eurasia, and also from North America, where we don't find the southern mammoth. And then later still, we have 2 other new mammoth taxa. We have the Columbian mammoth and the woolly mammoth. And the Columbian mammoth is only exclusively found in North America, whereas the woolly mammoth was found from Europe through Asia all the way to North America. Okay, so that's all very well. We've got 4 different mammoths. I've just explained like how they occur chronologically, but it's been a bit of a nightmare, like understanding exactly how they're related. And a number of hypotheses have been put forward based on morphology and also some earlier genetic work. And it's important to note that prior to our study, there was already a lot of genetics that had been done on the Columbian mammoth and the woolly mammoth. And there is very interesting, because some studies were suggesting that the Columbian mammoth actually fell within the diversity of woolly mammoth, which is very confusing because the anatomy of them is very, very different. So really, the way to kind of like get at this, to try and tease apart what is the Columbian mammoth, what is the woolly mammoth, how do they relate to each other, is to get some DNA from these really older, much older mammoths, the steppe mammoth. And that's what we did in our study. So we incorporated genomes that had already been collected from North America, which included Columbian mammoth and woolly mammoth and other woolly mammoths from Europe and Eurasia. And we included two of these old steppe mammoths, the Krestovka mammoth, which was the oldest at around 1.2 million years old, and Adichia, which was around 1 million years old, 1.1 million years old. And when we ran the ancient DNA on these and we reconstructed some genomes, we got some really interesting results, which is summarized in this figure. So what we find first is that the Adichia mammoth, so remember, these are two individual mammoths that should be steppe mammoth. The Adichia mammoth basically falls at the base of the tree, of the part of the tree that's leading to woolly mammoth. So we can say that Adichia belongs to a lineage that gave rise eventually to the woolly mammoth. But what's very interesting is the Krestovka mammoth is actually very different. It's very different. It's very different from both Adichia and woolly mammoths, and to some extent the Columbian mammoth. It's basically its own lineage. But what's very interesting is that the Columbian mammoth is actually about a 50-50 mix of this Krestovka lineage and the woolly mammoth, which would explain some of these issues that we were seeing with some of the earlier data where woolly mammoth looked like it was very similar to Columbian mammoth. If we kind of like put this kind of like these data into a cartoon, we can kind of like tell a story where at the base here we've got basically we've got the ancestor of mammoths and Asian elephants, which are the most closely related living animal to mammoths. They were living in Africa, and then they diverged within Africa. And then the first mammoth lineage that we know of was Mammuthus meridionalis, the southern mammoth. And then the southern mammoth split into these two distinct lineages, the Adichia and Krestovka. So these are the steppe mammoths. Now, as I said, we weren't sure if how steppe mammoths gave rise to woolly mammoths and Columbian mammoths. What we can say here is that steppe mammoths did give rise to both of them, but they gave rise to both of them in very different ways. So basically, the woolly mammoth is completely derived from the lineage that the Adichia mammoth belongs to, whereas the Columbian mammoth has this 50-50 mixture, this hybrid, is a hybrid species of the Krestovka lineage and the woolly mammoth lineage. What's very interesting is the fact that we can tell that this hybridization, this mixture event, occurred almost certainly in America, because that is the only place where we get the Columbian mammoth. So what we suspect is that the Krestovka lineage, so the steppe mammoth that belonged to the Krestovka lineage, first migrated into North America from Asia, and then later on, woolly mammoths also migrated into America, and it was there that the hybridization event happened. But this is all very interesting, because now we're talking about steppe mammoths. We've got two different lineages, and we've also got the woolly mammoth and the Columbian mammoth, which have a very, very different genomic history. How does the taxonomy relate to this? Should these two steppe mammoths be different species, or should all of these mammoths actually be collapsed to the same species? Which might be suggestive, because the Columbian mammoth is a hybrid and it lived for a long time. So these are kind of like outstanding questions in taxonomy that I know some of my colleagues are currently working on. Okay, so just to summarize, ancient DNA can survive for more than 1 million years, probably not for much longer, certainly no longer than at least 2 to 3 million years, because that's basically as theoretically old as the oldest permafrost is. And ancient DNA is really revealing this hidden diversity of extinct megafauna that you would never be able to find just by examining the bones using more traditional methods. We've shown that the stilt-legged horses actually probably their own genus. They definitely fall outside of the diversity of living horses and donkeys and zebras. And the steppe mammoths, we don't know yet. This is something that's ongoing to try and determine what's going on. What's very interesting is a lot of this diversity, it was all lost. And as I said at the beginning of this talk, we really want to kind of like understand how these things evolved, which populations went extinct, and also how ecologically diverse they were. And then this will inform the why. So basically, was it human hunting? Was it climate change? Or was it a bit of each? And then what we had were just ecological cascades, which basically just meant that huge swathes of the megafauna disappeared because they were reliant on one another. And these are questions that hopefully will be resolved in the not so distant future. And I'd just like to acknowledge the various researchers who basically were collaborators or led the studies for these two papers from all over the world. Excellent people. And if you want to learn more, this is the information on the two papers that I've talked about today. Thank you very much.
Wow. It was really an amazing presentation. Thank you, Pete, for doing that. It was really cool. I guess that we have some questions from the audience, right, Christopher and Linda?
Yes, thank you so much to both of you. I have a question, for both Pete and Sandra. And the first question is, what inspired you to study DNA?
Okay, I'll go first. So my background actually is in the paleontology side of things. So I did my master's in paleontology. And when I was doing that, as a part of that, I was looking at phylogenetics, so reconstructing family trees. And this is where I first got into the ancient DNA literature. And I found it was a really cool way at getting at some of these questions about how these extinct megafauna are related to one another. And basically, I got into ancient DNA to use it as a tool to basically get at some of these questions. And since then, I've kind of like diversified into sediments and various other things. So yeah, it basically really sucked me in.
Yeah, in my case, I come from a paleoecological background. So I studied a bit for my PhD, but so I was simply using pollen grains and diatoms, that is a kind of algae, to reconstruct the vegetation that surrounded that small pond and what was happening inside the lake. So all this, it's work that you do in the microscope. But when you are counting pollen, you can find much other organisms in there. So you can find acari or parts of fungi, spores, other things that you don't know what it is, but you know that it's organic matter, so it came from a plant or another organism, and most of it, like my DNA curiosity, arises from there. Like I could identify the pollen grains, but I couldn't identify all those organic remains that only one fragment couldn't tell me. But if I extract the DNA and I use the proper barcodes and small fragments, I can answer those questions. And that's how I came to DNA.
Okay, thanks. And then we will get into some questions. Some are short and some might be difficult. So yeah, and feel free, both of you, to answer. But the first one is, we have now seen how DNA can be used to look at diversity, both past and present. But is it also useful for the future? Can you sort of infer some knowledge for the future from this, or is it more of a historical context that it's interesting to look at?
Well, I think it has a lot of potential for the future, like one of the concerns about the climatic change and its effects on biodiversity, it's all these invasive species, as the last example that I showed. So if you use proper natural samplers, you can have like some sentinels about this species invasion and how to deal with them, because the impact for the ecosystems can be very high. And not only this, for example, regarding the part of biodiversity, like there are many species that are thought that are already extinct. But if you are also sampling DNA from the regions where where you think these animals are still there, then you can have like an indirect approach to know how those communities are dealing with, and if they are surviving, or you have to use another kind of measures to properly take care of them.
Thank you. And a natural question is it hard to extract this DNA? You talked about you found so much in the glass of water, and you find in ancient bones. How hard, how expensive, how time consuming is it to actually get this DNA?
Okay, so to extract the DNA is pretty straightforward, but unfortunately, until you've extracted it and done a bit of sequencing, you don't necessarily know if the DNA is actually any good. So it usually takes a few days to get from a specimen to extract the DNA, to then get the DNA sequences. And then it's usually a bit of trial and error to see basically which remains preserve well. But in general, if the remains have been preserved in permafrozen environments like permafrost, they tend to work pretty well. It's in more temperate environments so that it can be very hit and miss.
Okay, yeah, thanks. And well, I was naturally quite disappointed to learn that we can't get this Jurassic Park anytime soon. But how much information is contained in the DNA you actually can get? Could we theoretically have a perfect mammoth walking today based solely on the DNA, or doesn't the DNA tell the whole story?
Right. So in really well-preserved specimens, you can get really good quality genomes. You can get like high coverage, and you can look at what we call heterozygote sites. So you can look at basically sites, parts of the genome that are different between between the chromosomes. But there are certain parts of the genome which we just cannot reconstruct with current methods, because they're very just difficult to reconstruct anyway from modern DNA, but from ancient DNA they're near impossible for various technical reasons. And because of that, it's probably unlikely that we will reconstruct a mammoth, not to mention all the ethical issues and all the other problems that are associated with that.
It seems that we lost Christopher and Linda, so maybe I can just take care of the questions. So we have a couple of questions from the audience. One for Gerrit, that it's for both of you. So how long does it take to extract the DNA, and can you get how is the quantity of that? You are unmute, Sandra.
Yeah, you asked about what is the quantity of DNA that we can recover?
And how much you can do it.
And how
and how long does it take to extract the DNA? It's a very long procedure. It's short.
Well, with the kits and techniques that there are nowadays, it can be a very fast procedure. So let's say that since you go to fieldwork, you can get the water samples and extract DNA, do the library prep and sequence all that DNA maybe in it's 1 week work.
Mhm.
So the procedure can be very fast. I think there are also other sequencers that you can already bring to the fieldwork itself. So while you are still doing the survey, you can start the process.
Mhm.
So I think also the technology it's giving the chance to do things even faster and have cheaper and faster results.
Okay, good. Good to know that. Okay, and we have another question, in this case from Lucilla. Hi, I was curious about the quantitative analysis of eDNA. How can we infer the number of individuals based on the DNA quantity, considering that it deteriorates in the environment?
Okay, I will take this. Then the quantitative analysis of environmental DNA, it's not yet there. So most of you also need to consider two things. One, it's where does your DNA comes from. So as I said, we are all living traces of DNA, but you won't find the same amount of DNA if it's like a degraded DNA that it's outside. So you have a cell, an animal cell that breaks, and that DNA goes outside and gets fragmented. So you have like small traces of DNA in, let's say, an aquatic system in the water. But you can also have the full cell. So you have all the organelles inside there, and your DNA quality is higher, and you have more replicates of those sequences. But anyway, so let's say that with that trace DNA, that extracellular DNA that you find floating in the water, that you cannot do a quantitative analysis of that DNA. So it's presence absence. You have it or you don't have the sequence. That's the inference that you can have. However, with that intracellular DNA, you might be able to do a semi-quantitative estimation. But I think that technique still needs a lot of calibration to be able to get to this quantitative analysis. But I think it will get there.
Okay, nice. Thank you. Gerrit is asking again regarding his previous question. He asked before if we can get quantity also, and he's asking quantities per species promotion of different species, sorry.
So, for example, I know I will get to one of the organisms that I know. It's diatoms. Diatoms are algae, so they have a lot of chloroplast. And then, but what we know so far from diatoms is that each species has different number of chloroplasts in them. So if you are taking one diatom that have 2 chloroplasts in that cell and another that has 10, the amount of DNA that you will find from chloroplasts will be higher in the one that has a higher number of chloroplasts. And if you already know that, then you can calibrate your DNA results according to this. But I think that there's still a lot of base ground research to be able to do this calibration. But I think there are a lot of things going on, so probably at some point we might be able to get a more quantitative analysis from that environmental DNA.
Okay, nice, cool, thank you. I have a question for Pete. Mm-hmm. I'm curious about mammoths. How does the genome of mammoths match with the genome of elephants?
Right, so it's very close. So like, you know, more than 99% of the sequence is the same. And this is very helpful to us because part of the way we reassemble these genomes is we have to map them to what we call a reference genome. So we take all our short DNA sequences and we map them to this kind of like this genome that we have as a reference. Our reference genome, the closest living relative of mammoths, is the Asian elephant. And thankfully, we have a genome of that. So we can use that to map all these mammoth DNA sequences, and that is basically how we are able to do our analyses. So yeah, it's close enough that we can do that. But as for kind of like sequence divergence, it's more than 99% identical.
Okay, quite similar.
Yeah, very similar.
Yeah, and I'm curious about the diet of the mammoths. How is the diet, and how it differ from race, from the location?
So that's a good question. I know some people have done actually done ancient DNA analyses of coprolites of paleotheses, and also of stomach contents of woolly mammoths, and they've shown that. So from their teeth, we can tell from the scratch marks on the teeth from what they were eating, that they were eating a fair amount of grasses. But some DNA analyses are also suggesting that they were eating a lot more, also a lot of broad-leafed, what we call forbs, basically small flowering plants, which were also on the landscape. So they were probably eating a mix of things, and of course they probably would have been opportunists to a certain extent. But based on pollen and the teeth, it was more grasses. But yeah, okay. So there's a bit of conflicting information there. And as for the different, like the Colombian mammoth versus woolly mammoth, I'm not actually sure. I know a lot of the diet stuff has been done on the woolly mammoth because obviously the DNA preserves much better in the permafrost compared to where the Colombian mammoth is found in the more southerly parts of North America. So.
Okay, yeah, interesting. I have a question actually, not regarding the mammoth. It's related with science fact which you shared with us days before, and we published in our social media, and it says the science fact: camels first evolved in North America around 40 million years ago. They lived there continuously until their extinction in North America only around 12,000 years ago. And one of our followers, Joanne, hey Joanne, thank you for the question, asked, could men be responsible for that extinction?
Absolutely. No, human hunting causing the extinction is a prime candidate. As I said in my talk, it's not the only candidate. There was a lot of other things happening, primarily big major swings in climates that were fairly unprecedented in the last few 100,000 years. But what's really telling, and this is true of the horses as well. So horses, North America is one of the first places the horse family evolved in around 50 million years ago as well. And they lived there continuously until around 12,000 years ago. And the horses that live in North America now, they are all introduced by the Spanish in the 15th, 16th century. So yeah, it's very interesting that you've got camels and you've got horses all disappearing at the same time. And it is very coincident with humans moving. And we know that humans definitely did hunt both of these taxa because there have been archaeological sites that have been found with cut marks on camel bones and on horse bones. So we know that people were hunting them, but whether that ultimately caused their demise is still a kind of like a part of the debate. But yeah, it's certainly possible.
Okay.
Yep.
Yeah. It makes sense. Well, Christopher is asking, Pet, how much bones do you have at home?
So I should say, as someone who works in a museum, I don't have actually a private collection of bones. I keep bones in museum collections. So in the office in the museum in Tromsø, we've got a few bones. But actually, where the material I talked about are collected, they're actually in museum collections, either in Russia for the Siberian mammoths, or for the horses, they're in government paleontology, publicly available paleontology collections in the Yukon Territory of Canada. And that's where all these bones do belong, is in museum collections where they're open to study for everyone.
Okay, thank you. I guess Christopher will be a little bit disappointing now. I have a question for both of you, but maybe Pet, sorry, can answer, and later Sandra. you both are focused on the ancient, on the DNA of animals, but what about the human DNA? Can we look further to our DNA to check for family network? Are there any way or tools or online platform we can do it? Do you recommend any trustworthy tool?
Okay, so I'll just answer that first part of that question of like just the ancient DNA is used for humans. So I talked about megafauna, but there's a huge part of the ancient DNA community which is concentrated on archaeology and reconstructing the population movements of past human populations, and not just what we call anatomically modern humans, basically us. But also extinct close relatives like Neanderthals and Denisovans. And there was actually ancient DNA analyses which demonstrated that all people with ancestry outside of Africa have a component of Neanderthal ancestry. And some people even have Denisovan ancestry as well. And all of this was learnt from ancient DNA. I'll let Sandra answer the question about family trees.
Well, so far there are some companies that if you send them a fruitage of your saliva, they can extract DNA and then find the nearby heritage that you have, and also where part of this gene part comes from the Europe. So if you have a higher Asian influence or from Norway or from Nordic countries, like there are several of them that give these facilities.
Okay, well, nice to know it. I have a last question for both of you to finish this interesting talk. Is there any special field you'd like to investigate in the future? Well, always regarding DNA.
So I talked about in my talk, I talked obviously about looking at extinct populations and diversity based on bones, but as Sandra talked about from the modern environmental DNA perspective, you can actually use ancient environmental DNA. So you can extract DNA directly from cave sediments, from lake sediments, even from marine sediments. And you can use this to actually understand how diversity has changed through time at a really fine time scale. You're not reliant on finding bones because you can just look at basically sediments and mud. And that's an area that actually both of us work in at the moment. And it's a really rapidly expanding area, and it's hugely exciting and has a lot of potential to clarify a lot of questions.
Okay, nice. And you, Sandra?
Yeah, I completely agree with Pete. Like, I am fascinated with the way that you can answer many questions with the actual ecology relationship, like the example I show with the pollinators and plants. Like, if you are able to understand these relationships by nowadays, at some point you can bring them to the past and then better understand how all the changes that occurred, why did they occur, and by using the proper markers and with all these big data reference libraries of all the sequences of all the organisms that are being sequenced, this gonna give a lot of insights into these present and past ecological interactions.
Okay, it seems that DNA carries a lot of information, so there is much more to study. I will invite other volunteers, Jaume and Christopher and Linda for the moment had a problem, but just to give you a big applause. Thank you.
And sadly, we reached the end of the
festival here in Tromsø. I was just
to remember that you can still just watch the videos on the channel, And tomorrow there are a lot more evenings in the Pint of Science Norway. As well, if you are interested of being part of the festival, you can just contact us in the banners that appears here down, like our email and our webpage, to be both speakers or volunteers. So yeah, you can put. Well, it's not here. It's a sad that it's not here. Christopher and Linda. Well, they are at least Christopher coming, so you can. Excellent. So yeah, all the volunteers, Christopher, Linda, Fiori, and me, Jaume, want to thank you for listening to these great and inspiring presentations, and see you next year.